Hi everyone, welcome to Unit 3, Chapter 2, where we will be learning about inheritance. As always, if you click on this book, you will get a link to copy the guided notes, which you can use to take your notes and then turn in later for extra credit. So today we're going to be going over certain traits that are inherited from the gametes that we learned about last week. Last week we learned how the mother donates one gamete and the father donates the other gamete to produce a zygote. So humans have 46 chromosomes and they always come in pairs like this. One half from the mother and the other half from the father. So one chromosome will look like this, and they're always paired up. Now, there is a location on each chromosome, known as a loci, or the location of a given gene. If I look at this loci on the maternal chromosome, it will be the same gene that's on the same loci of the paternal chromosome. Now, just because it's the same gene does not mean it codes for the same trait. So for instance, if the gene has the dominant allele or the dominant gene, we may express the trait brown eyes. But if the recessive gene, same loci, nothing has changed about the location, but that gene now codes differently for the recessive type, we might appear to have light colored or blue eyes. This is the difference between the genotype and the phenotype. Remember, you get one gene from your mother and one gene from your father. So for instance, your genotype, you may have received a recessive gene from one of your parents, but since you received a dominant gene from your other parent, the phenotype would be expressed dominantly, in this case, brown eyes. I remember phenotype because it is the physical trait. The genotype is the gene that it codes for. And those are going to be located on a specific loci of a specific chromosome. Now, humans, we have 46 chromosomes, 23 from mom and 23 from dad. Now, human gametes have one sex chromosome and 22 autosomes. So that means that we're going to have 22 pairs of normal chromosomes, and then one sex chromosome from the father and one sex chromosome from the mother. Now, we are always going to inherit at least one X. Since the mother can only donate an X, her gametes will always have X. So the offspring will always have at least one X. And the father will determine whether that offspring is male or female. If his gamete has an X chromosome, the offspring will be female. If his gamete has a Y chromosome, the offspring will be male. Now, notice that the X chromosome is very large compared to the Y chromosome. That's because there's really not many traits that are coded, or there's not many genes coding for specific traits on the Y chromosome. The X chromosome is doing most of the heavy lifting. So a lot of the genes that are inherited on the X chromosome are on the sex chromosomes is located on the X chromosome. Now, females have two. That means one of these is pretty much useless, and we'll talk about that later. But here we can see the paternal side. You may inherit a dominant gene on this allele and a recessive gene on this allele from the maternal side, and we would express the dominant. Here we have a recessive allele and a recessive allele, so this offspring would express the recessive phenotype. Let's look at that again. If I have an allele for, let's say, the B trait, I'm not sure what this trait is, but B. I have this capital letter B. This represents a dominant gene. This capital letter B represents a dominant gene. So together they have the same dominant gene, whether from the maternal or the paternal side. Since they're the same, we call it homozygous. Homo meaning the same, zygote or zygous meaning zygote. So the zygote has the same trait coded on both of these loci. This is known as true breeding, because if I crossed a homozygous with a homozygous, all of the offspring would also be homozygous. In this case, it is homozygous dominant. Whereas over here, we also have a true breeding pair, but it is little b or lowercase b, which represents the recessive trait, and lowercase b, which represents the recessive trait. So it is also homozygous because the zygote has the same allele, but it is recessive. So this is homozygous recessive, whereas this one is homozygous dominant. Now, in normal autosomal inheritance, if the offspring is heterozygous, meaning we have one dominant and one recessive, it will only express the dominant phenotype because the recessive one is kind of covered up. So looking at these flies, this question asks what genotype is present with the phenotype normal wings. So I can look at these 4 flies. This fly has the phenotype normal wings. This one has normal wings, and this one has normal wings. The last one has the phenotype wrinkled wings, so I'm not going to pay attention to that one. Looking at all of these flies, they all have a dominant allele, one large capital W. These are heterozygous and this is homozygous, but the dominant allele is present, so I can assume that having normal wings is a dominant trait. So the genotype that's present has to have at least one dominant or one capital W. Looking at the same picture, it says what phenotype is present with the allele homozygous little w. So I'm looking at the genotype, and I want a homozygous little w. This one has 2 little w's. My phenotype is wrinkled wings, so I can safely assume that this is a recessive trait. Now, the only reason that we are able to know this much about inheritance is because of what we studied last week. The field of cytology is the field that studies cells, and cytologists were able to figure out how gametes were formed, or basically they figured out meiosis. Once we figured out meiosis, that paved the way for Gregor Mendel to be able to understand heredity. Here is a portrait of Gregor Mendel, and he is famous for studying pea plants. He just kind of happened upon the pea plant, but it was really, really lucky because the pea plant has several things going for it. Not only are there true breeding varieties, meaning varieties that if I cross them with themselves, I know are going to have the same reliable trait over and over again, but it also is a plant that can fertilize and reproduce very quickly. And I can cross-fertilize it, meaning I can take one of these true breeding green plants and true breeding yellow plants and get a variety of offspring. So I can track those genes and track those traits. So Mendel came up with something called the Punnett square, which allowed him to track these traits very reliably. What he did was he put the mother or the female gamete on the top and the male gamete on the bottom, and he wanted to kind of estimate how many of the offspring would produce which traits. So when he took a heterozygous allele and crossed it with another heterozygous allele, he had a 25% chance of having homozygous dominant and a 25% or 1 out of 4 chance of having a homozygous recessive. Then he also found that there was a 50% chance of getting a heterozygous dominant. Or if he did not know what was happening here, he could easily see that he got a 75% or 3 out of 4 of the offspring had the dominant phenotype. So if we were to identify which one was homozygous recessive, it would be this 25% chance right here. Homozygous dominant would be this 25% chance here, and heterozygous dominant would be a 50% chance right there. What if I did not cross a heterozygous gamete? So if I had one heterozygous and one homozygous gamete, it would change my Punnett square. If I pull down the A this direction, I know that at least 50% of the offspring is going to have a dominant phenotype. And then if I pull this one down here, I have recessive, and I pull this one across, I have recessive, and this one across. So I end up with 50% heterozygous dominant and 50% homozygous recessive. I do not have any homozygous dominant in this image. We can do it again. Now I have a dominant, a true breeding dominant allele at the top, and then a heterozygous dominant allele at the bottom. That means that I know for sure that 100% of my offspring will express the dominant phenotype, but 50% of them are heterozygous because they inherited the recessive allele from the father, and 50% are homozygous dominant because they inherited the dominant allele from the father. Let's look at eye color. Now, eye color is multi-allelic, meaning there's multiple genes that go into controlling this, but if we break it down to a really fundamental level, we can talk about blue and brown eyes. Now, if I have two parents that have brown eyes and I do not know the genotype, I only know the phenotype, I'm going to have about a 75% chance of having brown-eyed offspring and about a 25% chance of having light-eyed offspring because I don't know what the genotype behind this phenotype is. I only know for sure that the parents have at least one dominant allele. All of this changes when we're talking about two recessive. If we have two blue-eyed parents, I know for certain that both of those parents are homozygous recessive. So there is a 0% chance that the offspring would express the dominant trait because it's just not available. So let's see that in action. If I'm looking at this image, I have a blue-eyed parent. So I know for certain that I'm going to inherit a recessive allele from this parent. So all of the offspring will have at least one recessive allele. This parent is brown-eyed but is a carrier of the blue-eyed gene, meaning 50% of the offspring are going to inherit the dominant allele and express brown eyes, but they are heterozygous. 50% of the offspring have a chance of inheriting the recessive allele, meaning they will be blue-eyed. So this couple could potentially have a 50-50 chance of having a blue-eyed child. Whereas this couple, even though they are also blue-eyed mother and brown-eyed father, this brown-eyed father is a carrier for blue eyes, and this one is not. So we have a 100% chance of having brown-eyed offspring because the father has two dominant alleles. But all of those offspring have a chance of having a blue-eyed child because they are all carriers of the blue-eyed recessive allele. Now let's look at this family. In this family, every single member of the family has 12 fingers and 12 toes. This is a condition called polydactyly, poly meaning many, dactyl meaning fingers. So they have multiple fingers, multiple digits. Each one of them has an extra pinky and an extra pinky toe. Now, this family, I can safely assume is passing on a dominant allele just due to the fact of how prominent it is within that family. So all of the members of the family express it, meaning I'm assuming that whether they're homozygous or heterozygous, they're gonna have this dominant allele. Now, autosomal inheritance is not the only kind of inheritance that is available. So this is very straightforward autosomal inheritance. This is autosomal inheritance. I can assume that the dominant trait will overpower the recessive trait. But that is not always what's happening. Sometimes there's something called incomplete dominance. Incomplete dominance means that neither allele is really dominant over the other. So for instance, I have a true breeding red flower and I cross it with a true breeding white flower, or a homozygous red flower is crossed with a homozygous white flower. In normal autosomal inheritance, I might assume that 100% of the offspring would be red flowers with a carrier of the white flower trait, just like in this picture. But that's not what happened. We crossed it and expected to get red flowers with white carrier, but we actually got pink flowers. That's because in incomplete dominance, we actually have a blending of the two traits because neither one is dominant over the other. And we end up with 100% of the offspring having pink flowers. If I did an F2 cross and I crossed these two flowers with each other, I might have a 25% chance of getting a homozygous dominant red flower and a homozygous recessive white flower, and a 50% chance of also getting a pink flower if I continued this cross. But what we're going to get is a blending of the two traits. There's also something called codominance, where both traits are dominant and both want to be expressed equally. So if I take a true breeding or a homozygous red flower and a true breeding or a homozygous white flower and cross it, I'm not going to get a pink flower. I'm going to get a flower that is red and white. So here, according to this cross, I have red-white flowers. And this can express in many different ways. It can express as patches, or speckles, or spots, or stripes. It can express in many different kinds of ways, but you can think of it as both traits are fighting to be seen in the phenotype. Now, if I took this flower and bred it, I had a 25% chance of getting a red flower from this offspring, a 25% chance of getting a flower from this offspring, and a 50% chance of also getting a red and white flower. So let's look at some of these things. Incomplete dominance is where we have blending. Codominance is where both traits are expressed equally. So let's look at this genotype here. I'm going to turn on my pen here. Let me just do a quick cross. So I'm going to do a quick Punnett square. I'm going to have a mother that has the A blood type, okay? And I'm going to have a father that has the B blood type. Just bear with me. If I pull the A down, I know that these offspring are going to have A. So I have a 50% chance of inheriting the A allele. And then I'm going to pull this across. So that means I have B inherited this direction. Now the last thing that I need to pull down is little I, and over. I need to pull over little I. So let's look. This offspring, 25% chance of having type O blood. So that's this one right here. Okay. I have a 25% chance of having type A blood. Ooh, I did not mean to do that. Type A blood. Okay. This one right here. Okay, I'm only expressing A. Then I have a 25% chance of getting type B blood. So this one right here. Okay, but I also have a 25% chance of getting type A, B blood, where both alleles are expressed. So what type of inheritance is that? Does that sound like incomplete dominance or codominance? Well, since both alleles are expressed equally, it is an example of codominance. So blood typing is expressed as codominance. Now let's look at this picture. Which image represents incomplete dominance? Well, we said earlier incomplete dominance is blending. So these cows, I have a white cow and a red cow, and it looks like both white and red are expressed. So that is codominance. If I'm looking for incomplete dominance, I'm going to look for blending of the two traits. So this would be the correct answer. Now, blending can have multiple consequences. For instance, in these mice, if the mice have a double dominant trait, they're going to be the wild type. It's going to survive just fine. If they're heterozygous for this trait, it's a blending, it's incomplete dominance, I'm going to have a yellow coat. If I get both recessive alleles, this would be a white coat. But this mouse cannot survive this trait, so this zygote or this allele is never, ever, ever born. So instead of having a 25% chance or a 1 out of 4 chance, That's not what happens. I end up with something like this, 1 out of 3, or a 33% chance of getting the wild type coat. And I have a 66% chance of getting a yellow coat. I have a 0% chance of ever having a mouse with a white coat, 0%, because it dies. So many traits are inherited this way with a blending of the two features, and sometimes the recessive can be very dangerous and cause the zygote to die, essentially. Now, let's talk about dihybrid crosses. So far, we've only talked about a single cross or one allele. We're focusing on one allele at a time. So the Punnett square has 4 squares. But if I'm tracking 2 traits at the same time, my Punnett square will end up with 16 squares. So in this case, I'm looking at round versus wrinkled. So one trait is the shape. And I'm looking at the color, yellow versus green. This should be a lowercase r, and this should be a lowercase y. I think that it auto-corrected to capitalize it when it's not supposed to be. So wrinkled is actually a recessive trait, and green is actually a recessive trait in this cross. Now, I can count the phenotypes. And if I had a heterozygous, a perfectly heterozygous cross, meaning the father is heterozygous for both traits and the mother is heterozygous for both traits, I will see 1, 2, 3, 4, 5, 6, 7, 8, 9 dominant for both traits. 1, 2, 3 dominant for shape but not color. 1, 2, 3 dominant for color but not shape. And 1 recessive for both traits. So I will always have a 9 to 3 to 3 to 1 proportion. But usually the questions are going to ask you how many, or what is the proportion of P's in this cross that have recessive for both traits. So I'm going to just be looking at this one, which is 1 out of 16. 1 out of 16. That's a 6, guys. So sorry. It's really hard to draw with this thing. Or I could just ask how many, or what is the proportion of offspring that will be green. So I have 1, 2, 3, 4. 4 out of 16. Okay, and you can simplify that and say 1/4 or 25%. So we're still going to work with it the same way that we worked with the regular Punnett square, where we're pulling down the traits this way and pulling across the traits this way. But we're going to be working with 16 squares. Now, how do I determine what goes up here? Well, we're going to use the FOIL method. In math, you may have heard of FOIL, which is first, outside, inside, last. So if I am crossing these 2 cats, I'm going to take the first of both traits. So I'm going to take the capital S and the lowercase b. Then I'm going to take the outside, which is lowercase b and capital S. Then I'm going to take the inside, which is going to be capital S lowercase b, and then I'm going to take the last, which is capital S lowercase b. So if I FOIL this cat, I actually end up with this heterozygous cat right here. This is your F1 cross. If I FOIL this cat, I also get a heterozygous. I guess this example is just kind of predictable, but it is going to, well, once we cross them, we get heterozygous. So let's do a FOIL of this cat right here, because let's just say that this cat is the father cat. We have some little sperms up here. So FOIL is going to be first. The first letter is capital S and lowercase b. So we're going to get capital S lowercase b. And then the outside is going to be capital S, capital B. Capital S, capital B. Then the inside is going to be lowercase s, lowercase b. Lowercase s, lowercase b. And the last is going to be lowercase b, capital, lowercase s, capital B. Lowercase s, capital B. And that's what they did. They just did it in a weird order. I'm not sure why they did. They put the outside here and the inside there. But we're going to be using FOIL. If you would like a better explanation, there is this video right here where we talk about FOIL. You can watch this. Let's go ahead and do this cross. It says use FOIL to determine the parent alleles. This parent has already been done for us. So the male parent, we took the first, which is capital T lowercase g right here. The outside, which is capital T lowercase g again. Then we took the inside, which is lowercase t lowercase g right here. And then we put the last, lowercase t lowercase g. So we got this one. Let's do the other parent. So the other parent, we have lowercase t, capital G, lowercase g. So we're going to take the first, which is going to be lowercase t, capital G. We see that, lowercase t, capital G. Then we're going to take the outside, which is lowercase and lowercase. So here we have lowercase and lowercase. Then we're going to take the inside, which is this right here, lowercase t, capital G. The last is going to be lowercase, which is what we see here. So our answer to this question is lowercase t, capital G. Let's complete this dihybrid cross. Now, most of it is filled out except for this square, so all we have to do is pull down these alleles and pull across these alleles. So what I have is capital A, capital B, pulled down from the mother, and now across from the father, lowercase a, capital B. So my genotype is a heterozygous A, homozygous dominant B. My phenotype is going to be dominant for both traits. Now let's talk a little bit about linked genes. Linked genes mean that there are genes that are very close to each other in proximity, and so they tend to be inherited at the same time. So for instance, if the antenna length is right next to the body, then these genes may be inherited together from the same gamete. So we're just looking at physical proximity. So for instance, which gene is most likely to be linked to antenna size? I'm going to look and I see that this one has a small antenna and this one has a large antenna. So I assume that the allele for antenna size is 0. So these are the most likely ones to be linked to it, just because they're close in proximity. And this is especially true with crossing over, because if we crossed over this little section here, it is most likely that we're going to grab both of these genes at the same time because they are so close to each other in proximity. For the most part, this is really important when we're talking about sex-linked genes, or genes that are on the X allele. So if we have an X and a Y, and we have a trait that is linked to the X chromosome, this male is definitely going to express this trait. In this case, mutant eyes, which turn white. So this male expresses white eyes. A female would have to have this trait on both X chromosomes to be able to express this trait. So we know that this is a recessive trait. This female is a carrier, but she does not express the trait. So wild type or mutant type, it can be linked to the sex chromosome. That's not always the case, but what we're going to go over is talking about it being linked to the X chromosome. And that is exactly what we found out with these fruit flies, is that if the female had red eyes, most of the offspring is going to have, or all of the offspring is going to have red eyes, even if the male had white eyes. But if we cross the F2 generation, or we cross this generation, then we will start to see that white pop up again. Okay, we're going to start to see it pop up again because we're inheriting it from the male and the female at the same time. So let's look here. According to this diagram, is red eyes sex-linked or sex-influenced? Well, we know that it's sex-linked because here we have a white-eyed female and here we have white-eyed males, or we have a red-eyed male and here we have red-eyed females. If the trait was sex-influenced, it would only be expressed in one gender. Sex-influenced has more to do with hormones and the development of the animal. So for instance, a lion's mane is typically only seen on males, whereas this could be expressed on males or females. But it is expressed disproportionately on males because males only have one X chromosome, whereas females have two X chromosomes. So looking at this cross, I have a male which has red eyes or wild type eyes, and I have a female that has two recessive traits. So she expresses the mutant gene. The only reason she expresses it is because she has both of them. So let's look at the cross. If we carry this X over, then we know that we are inheriting one recessive allele on both of these flies and one recessive allele on both of these flies. Now we have to pull down the father. The father has a dominant allele, so that means that this fruit fly, or 25% of these fruit flies, are going to be carriers but not express the mutant trait. So they have red eyes. This one is a carrier, but it does not express the mutant trait because it has the dominant allele right here. So it also has red eyes. However, all of the male offspring, they are not inheriting an X from the father. They are only inheriting the Y from the father. So they can only express the mutant trait because that's the only trait that they got from the mother. So all of the males are going to express the mutant type, or the white eyes. Let's see. The reason why we see such a disproportionate effect on males when we have an X-linked chromosome is because females have two X chromosomes and males only have one. That means we pretty much have a 50-50 chance of getting that affected gene here. But females, if it's recessive, they have to have it on both sides. And that is because there's really extra information here, extra genetic information. The female does not need all of the genetic information from both of those X chromosomes. They only need one. So that second X chromosome is kind of inactivated. Okay, it's still there, it still does some stuff, but it's for the most part inactive, and we call it a bar body. Call it a bar body at this point. So that second X chromosome, not so important. So when we're talking about X-linked traits, if it's a recessive trait, it's going to affect males more because males only have one X chromosome. If it is a dominant trait, it's going to affect females more, or it's going to affect females more because they only have to have one to be expressed. But if it's recessive, they have to have two to be expressed, so it's not going to affect females as much as males. We see this a lot even in color blindness. So we see this kind of X-linked carrier effect with color blindness. This question says, can a woman have color blindness? Well, if we look at this, this is an X-linked recessive carrier mother. So the mother has one of the traits right here on her X chromosome. The male does not. So 50% of the offspring might be male. So they're going to inherit the X chromosome from the mother and the Y chromosome from the father. So that means there is a 50% chance that he will inherit this chromosome, which this child did. And since he only has one chromosome, he is affected. The female offspring will inherit an X from the father and an X from the mother. If he inherits, or if she inherits the normal X chromosome from the father and mother, she will be unaffected. If she inherits the mutated X chromosome from the mother, right here, she still will be unaffected. She will not express this color blindness. She will be what's known as a carrier. She carries the gene, but she does not express the gene. Okay, so it does not look so far like there is an affected female. Here we have an unaffected mother and an affected father. He, much like this child, okay, has the affected gene on the X chromosome. So all of his daughters will be carriers. All of the daughters will be carriers. None of the sons will have the trait. None of the sons will have the trait because they inherit the Y chromosome from the father. They don't inherit the X chromosome from the father. Neither of these daughters have color blindness. They are just carriers. They're just carriers. They just carry the trait. But what if we had an affected father and a carrier mother? If we had an affected father and a carrier mother, we might end up with a daughter that is affected. And it does happen, it's just very rare. So this doesn't really show what I was describing, a carrier mother and an affected father. It actually shows an unaffected father and an affected mother, meaning she carries two mutated genes, which means all of her offspring will have the color blindness trait, except the boys will be affected and the daughters will be carriers. So all of her daughters will be carriers. But yes, women can have color blindness. It's just a lot more rare than males because females have two X chromosomes. This can also happen with muscular dystrophy and hemophilia. This is a lot of the reason why the royal family had a lot of hemophilia that only seemed to affect the male heirs, because the males were more affected and the females were more carriers. All right, one of the ways that we can tell what's happening with these traits is by drawing a pedigree. Pedigrees really help us organize and track genetic traits when we're not quite certain of what the genotype is, but we do know what the phenotype is. So generally pedigrees, males are square and females are circular. Unaffected individuals are white and affected individuals are colored in. Sometimes you can see heterozygous as expressed as like stripes, but not always. Usually it's either affected or unaffected. Usually we see it affected or unaffected. So we're tracking one specific trait. When parents have offspring, we just draw lines to the offspring and we'll label it by generation. So let's look at a few of these examples. If I'm looking at this example, I automatically am going to assume that this is a dominant trait just because it affects so many members of the family, kind of like the polydactyly example that we went over earlier. I'm automatically going to assume that this one is a recessive trait because so few people are affected. But because I know this person is affected, I can automatically assume that both of these parents had to be carriers of that trait. If nothing else is going on, there's no links or sex linked or anything like that, I automatically assume that these parents are heterozygous, because in order to have an offspring that expresses that recessive trait, I would have to have a carrier parent. Now this one, we know for sure expresses the dominant trait, but we don't really know what that second allele is. It could be dominant or it could be recessive. All we know is the phenotype is dominant. We can do the same thing here. If we assume that this is recessive and we assume that this is dominant, we don't really know what this is. We know that the parents have to be heterozygous because the only way that they could have a recessive offspring is by having that recessive allele being carriers. Now it gets a little bit more complicated when we look at these generations. Here we have one generation, two generation, the third generation. Now looking at this whole family, I noticed that this is a female that's affected, this is a female that's affected, this is a female, this is a female, and this is a female. Most of the affected members of this family are female. So I assume sex linkages because it obviously has something to do with gender, but since most females are affected, I'm assuming that this is X-linked dominant. Compared to this family where this is a male that's affected, this is a male that's affected, this is a male, male, and male that is affected, I'm going to assume that this is X-linked recessive, because where this female only has to have one mutated gene, or it could have 2 mutated genes and it would still express it, or male would only have to have 1 mutated gene. I'm seeing a lot more females that are expressing the trait, okay? On this one, if the female only had 1 mutated gene, it would not express the trait. So that's why we see much more males, because they only have to have 1 copy. If a female were to express it, such as this female, I know that she would have to have a double dose or double recessive, which is why so many of her sons are affected. In fact, I can almost certainly assume that this daughter and this daughter are carriers of this disorder. So if I were to match these, I would say this one is dominant, this one is recessive, this one is X dominant, and this one is X recessive, because so many males are affected here and so many females are affected here. Let's look at this. It says in humans, polydactyly is a condition where we have extra digits, and this allele is dominant. A person with either heterozygous or homozygous dominant will have the trait of extra digits. Determine the genotype of this person. So we want to know what this person's genotype is. Now I know that this person is affected and this person is affected. This person is not affected. If I know that this person is not affected, it cannot be heterozygous dominant or homozygous dominant. It has to be, or this offspring has to be recessive for both traits, because it does not express the dominant trait. So this one is going to be homozygous recessive. Let's look at the next one. Determine the genotype of this offspring. This is tongue rolling, which is a recessive trait. So we do not express the trait here. We do express the trait here. So we know that this is a recessive trait, and we know that this one is recessive. Now, if I have an offspring that is recessive, this one could be dominant, but we don't really know. It's expressing the dominant trait, but we don't really know what that second allele is. We do know for sure that we had to inherit one of these recessive traits from the father and one of these recessive traits from the mother. So we know that both the mother and the father at least have 1 recessive trait. Now this one, this little girl, does not express tongue rolling, so she does not have the recessive, homozygous recessive. She could inherit 1 recessive from either the mother or father, but she has to have inherited at least 1 dominant from either the mother or father. So I can safely assume that the mother here is heterozygous. So I know for sure that this mother is heterozygous because she has 1 offspring that is recessive. All right, don't forget to take your weekly quiz here. And don't forget to do the chapter laboratory write-up. Also, there is an extra credit opportunity if you complete the guided homework. Thank you so much for joining me this week, and I will see you next week.